me, and I was about 6 years old when India got its independence. And I can tell you how poor India was. I cannot find a word in English that can describe the poverty. Dirt poor. So that is where Govind starts. And he had another unfortunate hit. India got independence. There's a partition. His whole family literally had to leave everything behind, shattered lives, and they come to Delhi destitute. You cannot imagine that. And I had really very long association with Govind, other than maybe Tom. And it was a non-traditional association. I was an honorary postdoc or researcher in Govind lab. I was at MIT due to Tom, and he introduced me to Irving London, and and it turns out that Irving London, Tom Rajbhandari, and Professor Khurana, they were very good friends. And so I come back to the village, and I wrote those words for Govind, and those words represent the values that he picked up as a child growing up in the village. And the fruit never falls far away from the tree, and those values he picked up from his parents. And everybody has said all the nice things about Govind, and they represent the values that he picked up from the village in Punjab. Now, I am not going to, I want to keep time because there are other people who, yes. A lot of things Govind told me, and you have already heard about this guy, Gordon Shrum. I brought this picture, and one day he told me that I am extremely grateful to Gordon Shrum because not only he gave me a job, he encouraged me, he supported me. More importantly, he said he protected me. And while the last thing he said to me in this sentence, but that he was my second father. Next person I want to bring up, everybody has already talked about Esther, and what nobody knows, because I had this non-traditional relationship that I was never a postdoc, and I could interact with him at a somewhat more relaxed position, environment, although also I had this medical degree which allowed me some measure of observing Govind from a different point of view, listening to him from a different point of view. And So it kind of gave me a little bit different perspective of Govind. Extremely nice, extremely nice, extremely nice, thoughtful, kind, all the things that you can think of. But I wanted to know how this shy person got to know this woman. I mean, there's no chance that Govind asked for a date. So I asked Esther that one day, I said, how did you meet Govind? Man, the question nobody ever asked her. She lit up and she said, I saw this very handsome guy walking around in a youth festival, Budapest, Hungary. So I went to him, introduced myself, and spent all that day with him. And she said, I found him absolutely fascinating, interesting, as shy as he was. And so she said, we exchanged our addresses, and ever since we kept in touch, and here we are together. They were madly in love, believe me. And she took care of everything you could imagine. And she kept track of every one of us. And even the children, their wives, and every Christmas time, there was a card coming from Esther to everybody. I still have that last card. And she came to every Corona meetings, even when she had cancer, even when she was in a wheelchair. Total dedication there. I think I'm going to stop here. I have maybe someday at a different time, a lot of interesting things. Such fascinating personality, such a great person to grace this world. And so I'm going to stop here because we have people who are, there's a program next one. So I'm going to stop here. I already took extra 5 minutes. Oh, you can see that.
Thank you, sir. It was really very emotional and a wonderful memory. I request to our chairman to please continue 2 more lectures as we are running short of time and our speaker has to leave early. So I request to sir, please continue 2 more lectures.
Oh, I'm going to be
as be a chair.
Oh, all right. Thank you. But I don't have the. Oh, Michelle. Well, Michelle, it's yeah.
There's some confusion. I'll request Dr. Michelle only to.
Oh,
yeah.
Michelle, you are the chair.
Sorry about the confusion. Dr. Ranu has actually taken us to the whole new world, so there was that confusion. So we are starting, we are not going to have the tea break, and in the interest of time, we are starting with the next session, which is on plant biology, which will be chaired by Professor Michelle Lovin. I'll just briefly introduce you. So Professor Lovin has done her PhD in biochemistry from Queen's University. She was postdoc in University of Basel, and then she did her postdoc again from MIT, and she's presently an adjunct assistant professor in Queen's University. I request her to please conduct the session. Yeah.
Okay, thank you. Okay. Our next speaker in this wonderful field of plant biology will be Dr. Nagendra Singh.
Just mention his area of interest.
Okay, his areas of interest are genome sequencing and annotation of crop plants and microbes important for Indian agriculture, and provides support to plant breeders for molecular marker-assisted breeding. He's been the recipient of a number of awards, including the Queen Elizabeth II Award in 1988, National Biosciences Award from the Government of India in 2002, the Rafi Ahmed Award from ICAR in 2007, and the list goes on. And with that, I will pass it to Dr. Singh.
Thank you very much, Mr. Chair. And very good afternoon to each one of you. At the outset, I'd like to express my gratitude to the organizers for calling me here for this very, very interesting seminar, a symposium in the memory of Dr. Hargobind Khorana. Of course, we have grown up when we went to school. We just we heard about Dr. Khorana, as somebody said, when he won this Nobel Prize. That was the time when we were going to schools to matriculation. And it was very inspiring to know that someone from India, after C.V. Raman, he was the one who got the Nobel Prize. Of course, after that, so many other people also got. So that is kind of inspirational personality he has been for all of us. What I'm going to, since time is very short, in this presentation I'm going to give a brief overview of some of the work which we have been doing in India under ICAR about the genome decoding. And this is the outline of my presentation. Why to decode genomes? Of course, many of you know that. But I will just go through that, our starting initial decoding of some of the genomes of rice, tomato, and wheat through international partnership. And then one or two applications of that. Then we have taken up 3 different genomes, pigeon pea, jute, and mango. And these are the crops which are uniquely important for India with very high economic significance. But it's important for India mainly. So we have taken up these things and then how we have proceeded in that, and I will just summarize the progress in that. Of course, this genome word was coined by a German scientist called Hans Winkler at the time when we didn't even know that DNA. But his definition is still 100% accurate. It is the complete set of genetic information present in a haploid cell of an organism, including its protoplasm. So this is the same definition. It is true, and it is very complete and accurate. Why we have to do it? Because many people question, particularly in a country, developing country like India, with a lot of resources, limitations, that why you should spend so much money. It's very expensive to do genome sequencing. I have tried to put in just 4 points. It has applications right from the beginning for very basic application to the actual practical utilization in the translational research. The very first thing, when we have the complete physical map of the genome, of course Dr. Khorana was one of the team who cracked the genetic code, and he was the first one to synthesize the gene. And now we have come such a long way that we can have a complete map, genetic map of the chromosomes from end to end. And once we have that, then we can do a lot of interesting things through comparative analysis, compare different genomes and see that how these different species on Earth are they related to each other, and how they have the common origin, how far back they have the common parents. All those kinds of new questions, the Darwinian evolution, those can be answered. So this is a basic curiosity of all of us, that we want to know where we have come from and how we will be going. So it provides very interesting. We don't have time, but one of my students have developed an eukaryotic tree of life, taking about 100 genes which are common in all the eukaryotes, and you get very good, nice, beautiful tree of life. The second one, once you come down, you can use this genome information for characterization of germplasm resources. Now, germplasm resources are very, very important in the age of intellectual property right. We have been talking about this IPR, WTO, these kind of things. The nations or the countries or the people who have the intellect, they are very high in technology. They have that is knowledge, that is called wealth. So now we are living in a world where the brain waves are also protected as a wealth. But the vast majority of people in Asia and Africa and South America, which are very poor, they don't have any intellectual property. Only property they have is their genetic resources, or genome or the germplasm. So there is a biodiversity convention also to protect that. But how would you protect when you don't even know that you cannot fingerprint it? So once you have a genome information, you can have very high density fingerprint, and you can protect your germplasm resources. If any right you have, you can claim and argue for that. Then further down the lane, you can actually use this genome information to find useful genes from the germplasm. Because anybody will use that germplasm, only purpose is to get the genes. Because the genes are there in the germplasm, in the resources. So that is really actually property which becomes really intellectual property, which can be used for useful purpose. So those genes can be mined. Having a genome information is just like having these GPS maps nowadays. These taxi people have, they can go anywhere. So having the genome helps to navigate through the genome to find the gene. And actually, last but not least, the actual breeding process as a plant breeders, when we do this selection, that selection process itself is helped by having the genome information. One can use the genetic marker for the foreground selection of the useful gene and the background. So these are starting right from the very, very basic to the applied. Any amount of money is spent on a genome which is important, a species which is very important for the economy, you get many times more in return. Now, this is a little bit complicated, but I won't go through here in more detail. It's just that we have different phases. You cannot complete a genome in 1 year or even 2 years. Usually they take quite a long time. Of course, even with a lot of development in the technology, still it is a slow process. Of course, it has become affordable and cheaper. So there are many species are there where we don't have any genome information. A lot of horticultural crops, fruit and vegetables, medicinal, which are very, very important, but we don't have any information. Then we can go to the preliminary draft stage, where 60 to 70% genome coverage. Many of the genomes have been published just like that, 60 to 80% coverage. Then few genomes which have the advanced draft, but not the reference level, just like a tomato, sorghum, chickpea, etc. Then very few genomes which we have very high quality draft, high quality reference sequence like Arabidopsis, rice or maize. Of course this number is increasing, but it's still the genomes the higher plant genomes you can count on the finger how many have been sequenced. And the reason why it is so difficult, because of the size of those genomes. If you see that the microbes have got only about 5 MB size, and now there is a technique you can sequence within a week. But as you go, see the wheat here, which has got 17 GB, which is 40 times bigger than the rice genome. So this enormous size of the genome gives a very challenging task to assemble it, because we have to sequence small bit by bit and then assemble back it like a jigsaw puzzle. So if the jigsaw picture is very large and pieces are small, it is going to be very difficult. And that's why wheat, even arguably the most important crops of the world, with the resource-rich countries, still it is not sequence finished. It's only in the draft stage. We don't have a reference genome of wheat. India first started its genome journey through the Rice Genome Project. Of course, we were not partner in the Arabidopsis and Human Genome Project, so that way we were lagging behind at least 5 years through the international community, but we eventually joined in year 2000. Our center at ICR and Delhi University, Professor Khurana is here, Professor Tyagi was leading this whole program, and we took this chromosome 11 long arm. And this was a wonderful partnership. We got to learn so many things, and it was a really complementary partnership. And this sequence was launched in 2002 and took another 3 years to get it the finished quality. And it was published in Nature here. And this paper has been quoted more than 2,000 times actually. Although there were 2 more papers which were really preliminary drafts. And they were done in 2002, but this was published in 2005. But this is one of the most used plant genome paper for people who are working in this area. Now that sequencing was done, approach was clone by clone approach. We had to clone the DNA to sequence. Whether you have the 2-step cloning, like a large fragment clone, then a small fragment, or directly go to a small fragment whole genome shotgun. But cloning was necessary because you have to use the Sanger method of sequencing. So it was used in the plasmid. And then with our experience, we got the responsibility to sequence chromosome 5 of tomato, because by this time we had learned all the tricks of the trade. We established the laboratories. I won't go into the detail. But it was quite expensive, and that's why in the beginning, in the tomato genome, which another had the consortium of 10 countries, it was decided to sequence only 20% of the genome, only the gene-rich region and the euchromatin on each of these chromosomes, 12 chromosomes. And then one has to find those bags belonging to specific chromosomes. And as it turned out that while sequencing, we find that the bags are going all over the chromosomes. Bags belonging to one country goes to another country. And then in 2008, this next generation sequencing technologies came, the 3 independent technology. So this old method of sequencing, clone by clone sequencing, ended in about 2008. And it was found very slow and costly. This paper was published. And then in 2008, 3 new techniques came, 454, Illumina, and Solid, which avoided the cloning. We didn't have to clone the DNA for sequencing, because cloning was replaced by PCR. And as a result, it became much cheaper and much faster. So eventually we ended up sequencing about 90% of the tomato genome. And this was also a time when it became unnecessary to make a big consortium of the countries, because even a single country could sequence a big genome, because there was no need to have big partnerships. And it was not feasible also, because how will you divide the genome into many pieces? This paper was published in Nature in 2012. And in each of these 2 papers, in rice genome and tomato genome, there were 25 authors each from India. So our contribution was really quite intensive, because it was slow and steady things, and a lot of things, effort was required to reach that high level of standard to satisfy the international community. Wheat, I was just telling you that wheat is still not finished, and it is still possible to collaborate, because wheat is perhaps one of the rare plant species where you can separate each of the chromosome arms. We have a special kind of material called double ditelocentric, which was developed by the Ernest Sears in University of Missouri. For each of the 21 chromosomes, they are in 2 pieces. So you can do the flow sorting, and telocentrics are much smaller than the other chromosomes. So each of the chromosome halves were purified, and now we have chromosome arm-specific DNA and the back library, which the 16 countries around the globe, they shared. And then still, they published this draft, which is only 60% of the coverage, a couple of years back. So we are still waiting for the reference genome of wheat. But now we are into next generation of sequencing age. And then go to the next step. I will just give you one example here. The purpose of the genome sequencing is to do gene discovery, which can be used in for translational research to develop better varieties of crops, which can give you the higher yield or disease resistance or quality. So we picked up these three particular traits through forward genetics approach, assisted by genome information. This is this variety is a Jaya, the first one, which is the Green Revolution variety. Then we have the new material, which has got 400 grains per panicule, but they are not a variety, they are just a genetic material. We wanted to know what the gene which makes it happen. then special basmati quality and salt tolerance material. We took these three, and I will just give you only one example. This high grain number per panicule, we identified a QTL, a major QTL on the long arm of chromosome 4, and then by combining the different gene expression profiling, gene expression profiling methodology and genetic mapping, we narrowed down to just few genes in that region which are responsible for this high grain number. At the same time, we mobilized this QTL into 12 different backgrounds. They are the very highly popular varieties of rice, and in each of the 12 varieties, the tiller number was reduced, but the grain number per panicule was increased. And we have done the yield trial also. The yield also increases because of these. Most of these grains are actually fertile. They are well-filled grains. Now, with the same information, we have in our center, we designed a gene chip for genotyping on the Affimatics platform using the single copy genes. This was a special kind of approach which gave a very good insights into the evolution of the and domestication of the rice. And we found that some of the Indian rice varieties, groups like Indica rice and Aus rice, they have the closest wild rice relatives already present within India. So that showed that the rice has a polyphyletic origin. The Japonica type perhaps originated in the Southeast Asia or South China, but the Indica type and Aus type that originated within South Asia because they have the closest relatives there. And some of these, because there was a lot of controversy about this monophyletic or polyphyletic origins, and we found that there are mutations. The key domestication genes like the Satyring gene and red pericarp, and there are half a dozen genes which are very key to domestication. And it was claimed that there is a single origin of domestication because the same allele is present in most of the variety. But we found that there are other alternate mutations which also give the white pericarp and which are present in the Aus type of variety. So that was possible because of this high density chip which we had, gene-based chip, and there we could work out the haplotype. For each gene we had the multiple SNPs, and you can see that how many alleles are for that gene, and you can draw this relationship. Also we can use that chip for background selection. Like here is one of the examples of the variety I showed you. We have transferred this QTL here on the long arm. So this increases the yield by about 10 to 15%, and we recovered rest of the basmati background, which is the blue here. So that means you maintain the original quality, everything of that variety, and you can transfer just the desired gene, just like you can do any of those genes. You can also do it for the bulk segregant analysis and identify the QTL, very much similar to what you will do when you use the biparental mapping population. But this study actually, we worked out some of the methodology related issues, that the original population should be very large, and the number of lines you bulk, that also should be large enough, then only your results will be accurate. Now let us come to the main thing which the title says, that the genomes which are economically important for India. So we took up this PIGEN-P. We developed a consortium of Indian laboratories with, in partnership with ICRISAT under Indo-US AKI, but as expected, there was a very less interest from the US side because PIGEN-P is not important for US. So, but we developed this thing because, see, India is the largest producer and more than 90% is produced and consumed in India. So Indian government put this money, and at that time, by this time, we had learned how to use the next generation sequencing technology, and particularly this 454, which was easier to assemble through bioinformatics, and we published the first draft of the PIGEN-P genome in 2011. This was the first complete genome done entirely within India. And similarly, in Delhi University also did the chickpea for that, because chickpea is similar way, it is very, very important for India. So once we learn through international cooperation, then one can use those techniques for the translational purpose within the country. So then PIGEN-P was such a craft that there was absolutely no information available there. So here generating these resources help us to start mapping and tagging all these genes for dwarfing genes which help the green revolution, rice and wheat. And we found similar kinds of gene in pigeon pea also. And now we are developing these varieties which are under the testing. And then we made a chip in pigeon pea also. We made a chip for 62,000 SNPs. And this chip has been very useful for doing the mapping, all those things, applications. I won't go into the detail. Jute is another crop. India and Bangladesh. It's a very large volume cash crop for India and Bangladesh. Nobody else will put their money in that. So we took this one, and then we have again published the jute genome, and we mapped the gene for the fiber quality. So these are the two papers here. Then let's come to the last one, which is the mango. Mango is actually known as the king of fruit in India here and many other countries also, because it is a very large volume, $10 billion value commodity this one is. But we hardly export anything because we don't have much breeding work, not much work on the quality. But if somebody will eat the Indian mango, they will not forget the taste. They will keep on like to eating it. But how to make that commodity export worth exporting, we need to put a lot of science in that. Again, India is the topmost producer and consumer. Mango has originated in India. There is no controversy about this, because we have 70 million years old fossil record in Meghalaya. And then mango has gone from India to rest of the subtropical world under recorded history. You don't need science for that, it is history. That on which date it went to Southeast Asia, to China, Australia, Africa and America. So there is no such controversy. We have more than 1,000 varieties of mango of all size, color, taste and thing. And the detailed characterization has been done by the horticulturist, but there was no genomic tools and these things available. So we took up this challenge of sequencing the mango variety Amrapali, which is the first hybrid variety, which is in the orchards, mango orchards. But when we assembled this genome using the Illumina reads, which are very short reads, we find the genome assembly was much larger than the estimated genome size. And then we actually did the genome size estimation again by flow cytometry, and we see that there was nothing wrong with the genome size estimation. Then how the assembly is coming larger than the genome? And instead of that, the answer to that was that it has very high heterozygosity. So maternal and paternal genomes were assembling separately. Wherever the heterozygosity was more, you are having those countries, they don't merge. And that's why then you need to find another solution. And then the third generation sequencing came to our rescue, which is the PacBio sequencing, which is a single molecule sequencing. You don't need to do even PCR, you just need only the DNA and sequences. Now the Oxford Nanopore is also coming. So here, because of the very long reads, and we generated the additional actually reads for that. So time is short, I'm just summarizing. We now have got this assembly, which is 403 MB long, which is almost 95% coverage of the genome. And it was possible to assemble this heterozygote genome because we can now go for very large overlap because of the longer reads and allow too many mismatches, which takes care of the heterozygosity, which is not possible with the short reads of Illumina. So as a result, we have submitted this sequence, and the genome coverage is very high. This genome was anchored using a map published by China. They published a very high density map of more than 6,000 markers in mango, and this was used to anchor the genome. All the 20 chromosomes have been anchored like this, and we have predicted 46,000 genes, and we find that the highest similarity is with Citrus sinensis. So the sweet orange has got the highest similarity with the mango. One is sweet, and then we can draw up the phylogenetic trees and all those things. Routine things, we have annotated the repeat elements in that. You have DNA transposon, simple repeats, low complexity, the non-LTR. Then we have annotated telomere, centromere, and non-coding RNA. Then we have found segmental duplications in that. Like many other genomes, the mango has also gone through the segmental duplication, it's an ancient polyploid, and you can date all those duplications when they took place and what relevance they have. The heterozygosity was worked out is very high, and every 38 base pair there is an indel or SNP. That was creating problem for the assembly with the short reads. Now again, we have created a chip for mango also, now this time 82,000 SNP. So all these resources, tools are going to be very useful. This one example here, the pulp percentage. Like the seed will be small and there will be more pulp. We found 3 major QTLs here, which is mapped by association mapping with very high lard value. So second last slide is that. Now what are the future challenges and opportunities? Now we have the genomics technology coming up very fast, is getting cheaper, and more development is taking place. We also have the optical mapping based on the single molecule optical mapping. So we can use these technologies to create the reference maps of the species which have got very high significance for India or for any other country who have who can put their money. Then we combine these things, single molecule technology now. We don't need to go back to these older technologies. Then also ISO-seq, their technology, you can have full-length RNA transcript, or splice variant. All those things can be combined with the high genetic map. So I just have given you these 3 examples, but what I'm trying to say, that similar thing we can do with the other species also. We have many plants, fruit, horticulture, and medicinal plant. This thing can be done, and then one can make use of that in developing better variety or preserving our germplasm or to IPR, and then identify new genes and markers, which is the ultimate purpose, that all this science should serve the society. Whatever science great science we are doing, so with the vision of Dr. Khurana, that unless it is serving the society, it is of no use. So we hope that I have given you the glimpse of what we are doing, and in what is the future, what more things we can do. Maybe young people will do. There are so many things to be done, and the time is the limiting factor. So I will stop here. Thank you very much for your attention.
Thank you very much, Dr. Singh. Do you have time for any questions?
Yes, if there are any questions, I'm happy. No question?
Any questions from the audience? Okay.
I can ask as many of the QTLs you have already identified in rice also. Some are responsible for salt tolerance, some are for yield also. So the genes which are actually there in the markers has also been identified?
In some cases, yeah.
But
in most of the cases, genes are not identified.
So that work is still pending.
Yeah, but we can still use it in the breeding, because if it has narrowed down to very small segment of maybe 200 kb or 300 kb, we are using them in the breeding. For drought tolerance, we have almost half a dozen QTLs. We have transferred in these high-yielding varieties, and they are now drought tolerant. Submergence tolerance gene is known. Salt tolerance, some genes are known. So this is the stepwise we have to proceed. Even before knowing the gene, we can make use of that.
Any other questions? If not, perhaps we'll carry on with our, thank you, Dr. Singh. Will you join me in thanking him again? We'll carry on with our program. Our next speaker is Dr. Jitendra Khurana, not Khurana, who obtained his PhD in botany from the University of Delhi in 1982, and he did postdoctoral work at the prestigious Smithsonian Institution in Washington and at Michigan State University. He returned to Delhi to join the newly established Department of Plant Molecular Biology at South Campus in 1988 and has been there ever since. He'll be telling us today about some interesting gene targets in plant development. And I think it's rice development.
Thank you very much. I must thank Dr. T.R. Sharma for giving me this opportunity to be a part of this very interesting symposium. I must also clarify, this is the same surname. Okay. It's not mine. Okay. So this is spelled differently. In fact, more than 99% Khurana is spelled U, not O. But then Hargobind Khurana was different. in many ways. Okay. Well, as Dr. N.K. Singh said, that some of us really grew up listening to the contributions of Hargobind Khurana. And interestingly, during my undergraduate days also at Delhi University, during our exams when we were taking practicals, whether that was in anatomy, physiology, morphology, rather than asking questions in that area, everybody will ask me, the teacher, what is the contribution of Hargobind Khurana? So this all went through almost from 1970, 71 to 75 till I did my master's. And in fact, that's what also motivated me to take up science as a career, although in plant sciences, not in medical sciences or other areas where Dr. Khurana worked in. But this is interesting that when I moved in at Michigan State, and also before that at Smithsonian Institution, I moved on to work in an area where one was interested in finding out how plants perceive light to regulate their development. So in that sense, I think later part of Dr. Khurana's contributions or adoption, in that sense, I think relates very well in the area of photobiology and signal transduction, the area which I also work on, although in plant systems. Well, most of you in plant sciences background know that most of the contributions, understanding about plant development has come through work on Arabidopsis. And as Dr. N.K. Singh mentioned, now rice is a model system for monocots or cereal crops. And in my lab, the focus has been on whether these cereal crops are developing in a different way than dicots like Arabidopsis, and whether some of the genes may have acquired novel functions to regulate their development. So I'm going to just be very, very focused on trying to focus on one particular transcription factor, which is a basic leucine zipper. And this is the one which is, So this HI5 is a basic leucine zipper transcription factor which was identified in Arabidopsis many years ago, and has turned out to be a very central regulator to downstream to many different kinds of receptors in plant systems. So the work which I'm going to be describing in today's presentation is done largely by Naini and Akanksha. Other people have done other work which I don't have time to talk about. Well, since some of you are from the area, you may not be familiar that how exactly plants perceive light. So I've just 3-4 slides to make a general introduction. In the last 50 years' time, it has become known that there are 3 major classes of sensory photoreceptors: phytochromes, which scan the red and far-red region of the solar spectrum, and blue and UVA part of the spectrum is sensed by cryptochromes and also phototropins. Now we also have some other sensory photoreceptors which are involved in biological clock, and they're called Zeitloops, and there are 3 of them known in plant system. Interestingly, UVB, which is known to be very, very harmful otherwise, also regulates some aspects of plant development, and now we also know what the receptor for UVB light is. This is something interesting that we also have cryptochromes in our own biological clock, and although they were discovered for the first time in Arabidopsis, but to our surprise, all the Drosophila also have cryptochrome, which are sensory molecules for controlling biological clock. But human systems, although it's a part of the biological clock feedback loop mechanism, but they don't sense light to regulate the biological clock. This is something very, very different in animal systems or mammalian systems. And so in that sense, I think it must have evolved some new functions in our own biological system. Phototropins are unique to plants, and in fact, I had some fundamental contribution to make here during my postdoc in Michigan State. I isolated the first ever mutant in phototropism, which later led to the discovery of this particular photoreceptor by Winslow Briggs at the Carnegie Institution in Stanford. Another thing I would like to mention also, somebody in the audience mentioned, or the speaker mentioned, about the work of Darwin in evolution. Some of you may not be familiar, and during the later part of his life, Darwin also worked on and also wrote a very famous book by the title, The Power of Movement in Plants. And he talked about phototropism, gravitropism, tendril coiling, things like that. And he was the first one to tell that the grass coleoptile, a seedling, bends towards light, and the light is perceived by the apical part, but the portion which bends actually is the subapical part of it, in a way that there has to be a transmission from the apical part to the lower part, which is going to be bending. And that turned out to be very interesting to find out that the first hormone auxin is actually involved in that. And so it has almost taken from 1880, when he mentioned about that, to 1997, when we had the first receptor which is the one which is responsible for sensing blue light to regulate phototropism, and that is what is phototropinase. Interesting part is that there are 5 phytochromes in Arabidopsis, 3 in monocots. There are 2 cryptochromes and 1 cryptochrome dashed. There are also 2 phototropins. So there are small variations within these systems, and they may also perform different functions. Okay, what exactly do these photoreceptors do? I'll just go through this particular life cycle of a plant. In fact, they regulate practically every aspect of plant development, but some of the major features which are listed here are, right from germination, there is a role of red and infrared light, and this is sensed by phytochrome to induce seed germination. Once a seedling emerges, if you allow a seed germination to occur in dark, you will have an etiolated phenotype, but when you are exposed to light, this actually undergoes photomorphogenesis and leads to chloroplast development, and further on regular development takes place. And this transition, which is called de-etiolation, is controlled by red and infrared light through phytochromes, by blue UVA light through cryptochromes. Now, another aspect which is very, very noticeable in terms of the response of light is, if you actually have a very dense planted crop or in a field anywhere where you plant, naturally the germination is not going to be very, very uniform. Some of the seeds might lag behind. As a result of that, the ones which are actually taken up earlier, they might shade the younger ones. And this leads to what is called shade avoidance, and this shaded actually seedling tries to elongate much longer. And this is controlled by red and infrared light through a specific class of photoreceptors called phytochrome B and phytochrome E. And actually this is a wasteful process, and if you are able to actually knock out that particular gene, you are able to have a very uniform cropping pattern as such. Now coming to phototropin, which is responsible for causing phototropism to a unilateral light, this is controlled by a phototropin called phototropin 1. And another phototropin which is also involved in regulating another aspect in plant development is the chloroplast movement. Every cell of a leaf has about 100 chloroplasts, and they keep moving. And what really happens is, if you have no light at all, they are uniformly distributed all through the cytosol. When they're exposed to some limiting light, they actually orient in a fashion so that they can capture maximum light for photosynthesis. But if you have a very high energy of light, this could be actually very, very damaging through photooxidation. They tend to run away from that and causes protection from that kind of high intensity light, and this is a response which is called chloroplast avoidance response. This is now known to be controlled by another phototropin which is called phototropin 2. Now this lower response is controlled by phototropin 1, but the higher one is controlled by phototropin 2. Now another response which everybody knows, and I think all biologists know about, stomatal opening closing, which is very vital for plant development, oxygen exchange, and other exchanges of the moisture also. This also is controlled by light, and within white light it is blue light, which is very critical for that. It turned out to be that all these responses were known to be controlled by blue light for almost 70-80 years. But FORT1 mutation was only affected in phototropism. FORT2 mutation only caused a response which was lacking in chloroplast movement. But then these 2 mutations actually did not cause any change in somatal movement as such. At that point of time, we thought that probably there is going to be a third receptor. But it turned out to be that both are essential for that. If you have a double mutant, that also will be missing this particular response. So now we know FORT1 controls phototropism, FORT2 controls chloroplast movement, FORT1 FORT2 together they control somatal movement. As I mentioned in the beginning also, this biological clock is entrained by light, and in case of plants specifically, we have a unique receptor called phytochrome, and like our own system, we also have cryptochromes. So both these photoreceptors sense light to entrain our biological clock, which is very critical also for transition to flowering, because that's the way these receptors sense photoperiod in the leaf and cause this transition to flowering. So both phytochrome, cryptochrome also are involved in transition to flowering, which is a very critical phase of life cycle, because without that we will not have any seed set, no harvest as such. So this is the overall background, giving an idea that how critical these photoreceptors are in regulating plant development all through their life cycle. Now coming to some of these components which work through downstream to these receptors. In fact, there are dozens of them, but I only listed few of them. So these are the receptors I just talked about. Downstream to them, there are these early signaling factors called PIF1, PKS1, PKS2. And downstream to that, I think this is one very critical factor called COP1, which is a component of E3 ligase involved in protein turnover. And downstream to that, another 2 important ones are Hi5 and H5H, which is a homolog of Hi5 in case of Arabidopsis. So this particular signal, I think, is turning out to be very, very important for all light signaling, whether it's in response to red light, far red light, blue light, or UVB light. Largely to the work done at Yale by Sheng-Wang Dang, now we know that this COP1 interacts with another component called SPA1. Together, they recruit Hi5. And once they recruit that, it's able to cause UV coordination, it will cause degradation. This all is happening when you have plants growing in dark. So because of this degradation of Hi5, there is no photomorphogenesis. So what is happening on exposure to light is, whether this is blue light or red light or far red light, this COP1 actually is shunted out of the nucleus. As a result of that, you don't have any interaction of this SPA1 and COP1, and Hi5 is free. As a result of that, this transcription factor is able to go and bind to the promoters of these genes and regulate gene expression, which leads to photomorphogenesis. You'll be surprised that from chip-chip analysis, Sheng-Wang Dang has shown Hi5 binds to promoters of about 3,000 genes to regulate photomorphogenesis. Some of them may be downregulated, others may be upregulated. Now, the work in our lab, so this is the overall background about our knowledge about light signaling, which largely came to work under Arabidopsis. At that point of time, as Dr. N.K. Singh said, their group and our group at South Campus, Delhi University, we participated in the international program to sequence rice genome. And so this was published in Nature in 2005. The issue was of August 11th. Incidentally, in the same week, Science carried a news that this sequence of genome of rice has been achieved. And in fact, they published this particular photograph where I happened to be there, although N.K. Singh was a partner, T.R. Sharma was a partner, my colleague was also a partner. Incidentally, this photograph was clicked when I was there attending a meeting in Japan, and they were not there. So this picture appeared in Science for that matter. But that is, I think they have equal contribution to sequencing of the rice genome as I had. Now, once this sequence became available, idea was to see whether how you actually utilize this particular information to identify genes and also their function. So as a 5-lab consortium, we undertook this particular project where we tried to understand genes which are regulating transition to flowering and panicle development as seed set. And 3 of our colleagues, Dr. Tyagi, Dr. Sanjay Kapoor, and myself, we took up this challenge along with Usha Vijayaraghavan in Bangalore. We try to identify genes which are differentially expressing during various phases of transition. So we took 3 as control, which were vegetative, leaf, fruit, and seedling. We decided to choose the stages from fuller transition, shoot apical meristem, and 4 of other stages along with that. 7 stages was chosen during panicle development when the transition already taken place. We also chose to work on 5 stages of seed development right after fertilization, how the seed matures. And this all data which came through was very, very remarkable. It gave us an idea that how during different stages of development there is a differential expression gene occurring. Every stage there was a differential expression, but there were major spikes during development. And what are these stages which are representing overall floral development? This particular major jump occurs during organogenesis, followed by meiosis, polymitosis, sperm cell differentiation, fertilization, embryo development, like that. So I think we came up with a very nice data which showed that there were major changes occurring in development of the panicle, and gene expression was getting highlighted during these stages. This particular heat map also shows that during panicle development stages, some of these transcription factors, their expression was changing during from P1 to P6 stage. And looking at this list, it looks like some of the major classes are of DOF domain factors, PhD, SRF, BZIP, Polycom, and of course PL family, homodomain, and also SRF gene family. So I'm going to focus only on this particular transcription factor family, which my lab analyzed at the whole genome level. Also analyze how differentially they are expressing during panicle development. This is one example here I'm showing. You can see some of these genes, BZ47, 62, 41. There are about 89 genes of BZ family in rice. And you can see that how the transition is taking place. They are not expressing much during vegetative tissues, but they are expressing during panicle development after the transition has taken place, and their expression goes down during seed development. And there are others which are not expressing at all in all these stages, but their expression is very, very high during seed maturation. This kind of expression, I think, was providing us a clue that they must be having a specific role to play during those stages of panicle and seed set in case of rice. Now, I'm not going to touch upon all these genes, but I'm only going to focus on now this Hi5, which I said is a basic transcription factor. And there's only one high 5 in case of Arabidopsis. So once the genome was available, we made this phylogenetic tree. It turned out to be that although Arabidopsis only has one high 5, it has one high 5 homolog. But in case of rice, we were able to identify 3 of them, BZ1, BZ48, BZ18, which were homologous to ATH5. So the question was, which of them is actually more close to high 5 in function, or whether they have some kind of overlapping function, or they have acquired newer functions during the course of evolution in case of monocots like rice. So we chose to work on one of them, and which is BZ48. And because this was easy to clone to begin with, that's why the work started in that direction much more than the other ones, BZ1 and BZ40 and BZ18. Now, first thing to show is whether is it functionally equivalent to high 5 Arabidopsis or not. Easy thing was to be able to complement Arabidopsis mutant with this particular gene. So look at this particular phenotype. This is wild type Arabidopsis, which is about 4-day old, grown in light. And this is that high 5 mutant. When you knock it out, it causes elongation in the seedling height. And now you put this particular rice gene and make the complementation, you can rescue the phenotype, make it wild type. So in this respect, it's functionally equivalent to high 5 Arabidopsis. And if you express this particular gene in wild type, it does not make much of a change. At the same time, we also realized that there were some other traits also in this particular mutant of Arabidopsis. Look at this particular seedling position of the cotyledons. They are actually in this particular orientation here. And in case of the mutant high 5, they are vertically bent down. And this phenotype also could be rescued by the rice gene. And this is another trait which was also rescued. And look at this phenotype high 5 and the wild type. Look at this, the angle of the fruit, which is called silicua, like custard plant. This has a certain angle which is very, very steep along the axis of the stem in case of wild type. But this is much wider in case of the mutant high 5. And we could also restore that by putting the rice gene. Although this gene and the mutant has been worked out for almost 20 years, but to our surprise, nobody noted this particular phenotype, and we were the first one to report. This is also something which is affected in case of mutation in high 5 and could be restored by Arabidopsis gene. Now, as I said, if you knock out this particular gene in Arabidopsis, it causes elongation, and is it in a way also involved in controlling plant height? Everybody knows that what exactly the contribution of Green Revolution was, and Norman Borlaug got the Nobel Prize for that. This was also a gene which was involved in controlling plant height. These were the natural wheat genotypes at that point of time, and because of the mutation it causes some kind of semi-dwarfism, which led to a lot of stability in the plant and also higher yield, which led to Green Revolution. So we are also keen to know whether this light-induced gene also, or lighter gene also may have a role to play in controlling plant height. So next slide will give you some idea. This particular gene now, OZ48, we were able to overexpress in rice. This is the wild type. And also these 3 independent lines of transgenics, which are overexpressing BZ48, A orthologo high 5. This causes drastic reduction in the plant height. But this is a surprising thing, because in case of high 5 in Arabidopsis, although when you knock it out, it causes elongation, but somehow when they overexpress that, it did not cause a reduction. And this is the change we are finding is happening in case of rice versus Arabidopsis. This is very, very dramatic reduction in the height as compared to other genes known in literature which causes regulation of plant height. We also were able to see that, of course, this is now the internode of wild type. Within the same height, we have 2 internodes in case of the overexpression line, which is causing dwarfism. And there were also some changes also in the overall girth of the plant, and also changes in cell size and also cell length, which is also estimated here. So these kind of changes, I think we thought should have been very, very useful to give it some kind of a sturdy phenotype of the plant. It turned out to be the other way around, because this particular section shows that overall girth actually of the stem has reduced, and it's causing some kind of brittleness. Okay, brittleness to the stem, it may not be very, very useful overall for plant strength and overall flexibility. When you knock this particular gene out, you also find that there is elongation of the internode. Not only that, you also find aerial roots coming out of that, which is also expected, like in case of Arabidopsis. Now this came as a surprise. We also got to obtain one tDNA insertion line, and when we had the homozygous line, it turned out to be seedling lethal. In case of Arabidopsis, it is viable. When you knock the gene out, it actually becomes seedling lethal, which also is something a unique observation we made in terms of this role in rice development. We also carried out microarray analysis of all these overexpression lines and also knockdown lines. and found out that there are different hormone pathways involved in GA biosynthesis, BR biosynthesis, jasmanic acid, or also ethylene, which are affected differentially. And you can see in heat maps showing that overexpression causes downregulation of these genes, and they are upregulated in case of controls. Now, once we sent the data for publication to Plant Physiology, they wanted us to show that one of the genes we claimed is downregulated is this KO2, which is involved in actually initial steps of biosynthesis of GA from this particular G-adenylyl pyrophosphate to GA12, and it is involved in regulating 3 steps of the hair, causing the formation of kyurinate, which finally goes to form GA12 aldehyde. Finally, you have active GA, GA1, and GA4. And we were able to finally show in the promoter of this particular gene, there are 3 light responsive elements. Out of that, one of them is the one which is actually binding to that particular high 5 and also show reduction in the movement of the fragment during our EMSA. And when we mutated that, they were not able to cause reduction in the movement, showing very critically that this high 5 ortholog in rice is able to bind to the promoter of KO2 and regulate the biosynthesis of GA. Maybe because of that, you have reduction in the plant height. Now, another question came, as I told you in the very beginning in the introductory slide of Xinguang Dang, this particular protein is actually degraded in dark and also is stable in light. That's why it's able to regulate photomorphogenesis. Question came, what happens to BZ48 in case of rice? Is it light stable or is it stable in dark or not? Now, here is an experiment which I think very clearly shows that this is now a 4-day-old seedling grown in light or dark and then exposed to dark in this case and move on to light for various hours. We did not find any change in its overall expression profile. These 2 bands show that one of them is actually a form which is phosphorylated, which is well known in case of Arabidopsis also. So this is in contrast to Arabidopsis, because in case of Arabidopsis in dark, when you transfer plants of Arabidopsis from light to dark for various hours, you find there's a degradation of this particular protein. And that's the reason why it does not differentiate in case of darkness. But then, as again from dark, if you expose it to light, you're able to actually recover its accumulation as a protein. Now, the question came from examiners again, who were reviewing our paper, that you have shown that in case of rice, this is a stable protein, both in light and dark. You have also expressed this protein in case of Arabidopsis. Is it stable there or not? Does it behave like Arabidopsis, or is it also behaving differently? So this is that experiment which shows very clearly this 48 gene expressed in Arabidopsis also is expressing a protein which is not again degraded in Arabidopsis. Now we have shown that this degradation occurs because of the E3 ligase, which is COP1. The COP1 in rice, in fact, does not interact physically with 48, and as a result of that, this protein remains stable. So this particular whole data which I presented is now due for publication. In fact, it should have appeared about in the month of September, but the editors of Plant Physiology advised that we have a focus issue coming in February next year, and with the title Energy, Focus on Light and Oxygen, whether would you like to put this paper in that particular issue of that, and we agreed for that. So all this available online, but it will be now the whole data I presented will be appearing in Plant Physiology, published by the American Society of Plant Biologists. And so in conclusion, I would like to say that although the Arabidopsis mutant we have been able to rescue using rice gene BZ48, but when you overexpress that in case of rice, we are able to cause reduction in height, which is in contrast to what happened in case of Arabidopsis. Also, in case of rice, when we knock this gene down, we have internode elongation. But then, if you go to next generation, we have much more expression of this, down regulation of this. These seedlings become lethal, and as a result of that, the plant is not able to survive. Observation again, which is in contrast to Arabidopsis. And this, I think, is a high point of this particular presentation here, that the protein high 5 is unstable in dark, but this BZ48 of rice, which is orthologous to Arabidopsis gene, the protein somehow is stable and might be causing some functional roles which are quite in contrast in contrast to Arabidopsis. With that, I would like to conclude, and thank you for your attention.
Okay, thank you very much. Any questions? Okay, I have a quick question.
Yeah, sure.
So I'm doing a wheat global transcriptomic analysis, and I'm thinking I'd like to add your target to my list of genes. Do you know if there's a homolog in wheat? Have you looked?
Yeah, there are.
So
Homelock in wheat also.
It's across the board because, of
That's
course,
right.
Arabidopsis is
Right.
monocot, but
That's right.
rice isn't.
Right.
Thanks.
Yep. Okay.
Any other question? Any other question? Oh.
So overexpression of BZ48 leads to the retarded growth in rice. So you have any insight into it except for the metabolic pathway? Because I feel that because since it's a BZ transcription factor, it is expected to have number of heterodimerizing partners, not as a homodimer. So probably what is happening is that you are overexpressing a protein,
Yeah.
and now it's just dissociation constant. It's so much of it's there that is going to interact with its partner, and as a result, is being sequenced out, and the genes are downregulated as a result. Is that a possibility?
Not exactly. It does form a homodimer. It does form a heterodimer with BZIP1, also with BZIP18, and we are testing others also. But what exactly is known is now, because when you talk about reduction in height, ultimately it boils down to control of hormone levels and hormone action. Ultimately, GA, auxins, all of those are going to be regulating that. So somewhere down the line, you have to think about that light is pursued by cryptochromes and phytochromes. They then turn on the activity of Hi5, and Hi5 eventually is able to go and bind to the promoters of all these genes. So looks like that overall plant growth and development by light is coming again at the level of hormone. And that also, you know also that GA also controls cell elongation as such, and some of these phenotypes I've shown also shows change in cell elongation. So I think there is a clear connection which is coming, and there is no point of any other deviation as such. So light regulates plant height by modulating these through these factors, the hormone levels, which eventually will regulate the cell size and cell elongation and controlling plant height. Yeah.
I have a question. Do you have any insight into how these proteins are reacting to light or responding to light? Does it involve the aromatic amino acids? Does it involve photooxidative modifications? Once they've responded, do you think they get degraded and recycled, or is it like in Bacteroidopsin where there's a conformational change which is reversible?
No, I think they are degraded. There is no recycling as such. But I think what light is doing, because of its prevention, there is some change in its phosphorylation status. So phosphorylation also is one critical factor.
But is that mediated by the aromatic amino acids because there's nothing else that would respond to UVB differentially? In fact, all proteins would have, there are 4 chromophores in proteins.
Right.
Unless they have something else, like hemoglobin or something. The peptide bond and the 3 aromatic amino acids.
Right.
So, and they're there in most proteins. Yeah. So what is it that allows these proteins to respond to light differentially?
No, actually, light is being perceived by phytochromes, cryptochromes. So they are all redox changes which happens in case of other receptors also. So these are the steps which are coming in signal transduction through kinases and all that. So basically those are the things which are causing phosphorylation also. So you have to bring in, I think if I go to the very second slide I showed you,
So
there are intermediate components which will channelize the energy. So light force is working with the photoreceptors at redox level, changes are there, and that photoactivation will lead to kinase getting activated. Those kinases will work on these proteins.
Cascades of phosphorylation.
That's right. That's right.
Profiling of hormones in the contrastive behavior happened in rice and Arabidopsis?
We have not done the estimation of hormones as such. So some of those genes which we found in the microarray, we confirmed that with the real-time PCR, but we have not done the estimation on the hormones as such. Yeah.
Okay, and I think we should probably, is that working? Move on to our next, that's not working. Move on to our next, sorry, thanks Dr. Khurana for that lovely example of classic plant molecular biology. So our next speaker will be Dr. Banerjee. And while he comes up here, everybody stand up for a minute, because I can just see it. You're all falling asleep, and so just stand up for a minute. 60 seconds while we get the next talk ready. But don't leave the room.
So we break?
Well, I'll introduce you.
Okay.
I'll say a couple more words. Okay, and so perhaps I'll even start introductions. Thank you, Dr. Singh. That was a good talk. Train, yeah. I'll start with an introduction. So Dr. Anjan Banerjee is from the Indian Institute of Science, Education and Research in Pune. He got his PhD in biotechnology at the University of Pune with research work at the National Chemical Laboratory. He did a postdoc at Iowa State University in the US, and he returned to India as an assistant professor at Indian Institute of Science, Education and Research in 2008 and has been there ever since. And he'll be talking to us about the role of mobile RNAs in tuber development in potato.
So, good afternoon, everyone. First of all, I'd like to thank the organizers, Professor Sharma, Dr. Pandey, and others for giving me an opportunity to come here and share with you the research that I do. So I'm really grateful. Unlike all my senior speakers, my lab is 9 years old. I have been working on this field of understanding RNA transport, mobile RNA. You must have by now read my title. The mobile RNAs play an activator repressor role to control tuber formations in potato. Potato is the third most important food crop, right after rice, wheat, or if you bring maize in between, then it becomes fourth. Whatever it is a very important food crop in terms of its energy, in terms of its bioenergetic cost. So we really pay a lot of attention to understand what this mechanism is and how this mechanism, how this plant really drive the, turn on the switch to make potatoes. So it's basically, Professor Khurana has described a lot of plant development, and I hope the stage is pretty set now for you to understand what is that I'm going to talk about. I'm going to talk about mobile RNAs and what are these mobile RNAs. Mobile RNA field is not very old, roughly about 10 to 12 years of old. From UC Davis, many people have done phenomenal work to show us that what mobile RNAs are. And this concept came to the field indeed in the community of plant biologists. People did not know what causes flowering. So we had an old concept of Lorigen from 1935. And 2005, it was first time people came back. Ove Nelson's group showed that it is the FT mRNA which moves, flowering time mRNA moves and causes a plant to flower. But this result was later disputed 2 years later in 2007, and then people challenged, and then it turned out that it is not a mobile RNA anymore, it's a protein. In last 10 years, this field has gone much, much ahead beyond our understanding that what are these mobile RNAs and do they plant. Why would plants select mobile RNA? And today I have glimpses of results from my lab that I would like to share with you. That in the life of potato, we clearly established that there are mobile RNAs. And the title I showed to you, the mobile RNAs play an activator repressor role. Means it activates as well as it represses. So why would that do? So try to understand. And I think I'll give you some introductions in the beginning for you to look at what these mobile RNAs are. So wherever I go, people ask me a number of questions, and I like to make it is easier to see. Let's look at those questions first. What are those mobile RNAs? Why do they need to be mobile? And do they move short distance or long distance? What is this movement associated with? How is this movement, how do they move? The big question comes to the field today, how do they move, right? Do they move alone? So these answers I'm required to give you today. Otherwise you don't let me go. Does all RNA move? Question I have to answer. Are they repressed from translations or protein synthesis? Because it's a very fundamental concept, DNA transcriptions, translations. You like to see an RNA make the protein, but not always. We have seen there are RNAs that don't really make proteins at the site of their transcriptions. So it's a very fascinating area emerging in the life of plants, and with respect to it is now basically redefining our understanding in respect to plant development, be it stress, be it defense, whatever you consider in the life of plants. Are they more stable than any other RNAs? And indeed they are. We will show you that is the case. But does small RNA move? Another level of regulator coming in, the master regulator today are the small RNAs, and we now know that yes, there are examples that small RNAs are indeed 2010 onwards, now we know that in the field of plant science that small RNAs do move in the life of in plants. So if you look at the whole plant level, this is nothing but, you can imagine that this is the shoot apex and this is the root, right? This is the leaf, these are axils. So if you look at all these response, Professor Khurana has described very clearly the light and its major role in terms of plant growth and development, the photoreceptors. So now, how does this leaf perceive the signal? And once it perceives the signal, does it, all the signals get translated to protein right up there, or there are RNAs, they go from here to either towards the apex or towards the down? That's the question. And that is all about the story of mobile RNAs, okay? So I hope I will be able to give you some. So this is what we say this is nothing but the super highway, because we have now seen that if you take a scan, if you take a cross sections, and if you take those cells which are called vascular bundles, those of you know vascular bundles, it has got phloem and xylem. If you take phloem, you will find plenty of RNAs are present in phloem. But phloem is indeed anuclear. There is no nuclear machinery in the phloem. Where those RNA come to the phloem? That means these RNAs are expressed somewhere in the plant and then they go to the phloem. Now, why would this RNA molecule go to the phloem? Do they have any job associated with it? Or do they have any functions linked to it? That's the questions we are asking, right? And I take potato as a model system to understand, and hopefully I should be able to convince you that what is this concept all about, okay? This is the source and sink. This relation is nothing but, as a plant grows from juvenile to the reproductive phase, it has its flower. It's a reproductive fruit. So fruit is a strong sink from the leaf to the strong sink. The food must go, or whatever, be its development, be its growth, be its defense, all sorts of molecules must transport from the source to the sink. That's what we say. So I will use this word source and sink. You will say that anything I talk about in source, that means this RNA must be expressed in the source, and this RNA must have transported through the phloem stream to the sink. And in potato, it is potato itself the sink. So all these RNA molecules, they move, unlike animal cell, they have gap junctions. In plants, we have plasmodesmata. And plasmodesmata is the connecting gate between cell to cell. So they move from one cell to another cell through this plasmodesmata. Again, this plasmodesmata has the size exclusion limit. Not all RNA can move. Not all RNA protein complex can move. We have now evidence of all this. Okay, so if you consider the plant, you can look at how is this. It is the plant vascular system functions as the conduit for long distance communication between distantly located organs. Organs located here from root to shoot, from shoot to root communications, the discussions, the talk that plant execute without our knowledge, okay? And that is what it is. So schematic illustrations of the xylem and phloem, illustrating the fundamental feature that in contrast to the animal kingdom, the flow pathway is non-circulatory in nature. So we are talking about this complete pathway inside the plant. This is a review. I give you a cartoon that how these RNA molecules are likely to move from cell to cell. I will again bring out the evidence later. But for you to understand, RNA is a long distance macromolecule. And this is cell autonomous RNA. I say CAR versus NCAR because RNA molecules are transcribed in a particular cell and it goes somewhere else. So that's a non-CAR, NCAR. That means it is non-cell autonomous transport, and there is a cell autonomous transport as well. This is the model organism that I use to understand this concept. If you look at this model organism, you see these potatoes are grown below ground. This potato, by the way, it's a modified stem. Now the first question comes to my mind, it's a modified stem. Why would plant grow a modified stem below the ground? Why doesn't it grow above the ground? Right? So I have an answer. If you can answer, that will be nice. So this is a modified stem which grows. You can look at this stem, how this stem grows below the ground, and it swells up. And this swells up, this organ ultimately develop to a potato. Completely reloading and loading and reloading of sucrose from the phloem after photosynthesis, it just stores in this organ and makes the big organ for us to understand. Okay, if you look this slide cartoon, it will be much easier for you to understand even better. You can look at this. This is this organ. Look at this transition of this organ. Okay, from this organ it develops it's a hook-like structure. From this hook-like structure, it gradually swells up, right? And this swelling basically allow sucrose to accumulate, photosynthate to accumulate. Lot of cell division changes, lot of cell fate changes, lot of developmental changes happens in this organ, and finally this makes a big tuber for us. Okay. This phenomenon is known as tuberization. We tell them it's a tuberization, and this is governed by number of factors. Okay, so who are these factors? I made a cartoon slides for students to understand. So the players like nitrogen plays a critical role. Photoperiod, the light duration, short day versus long day. Photoreceptors, of course, phytochrome. Temperature also plays a major role in these transitions of this organ from here to here. Endogenous factors, you can talk about hormones. You can also talk about small RNAs. We now know small RNAs also play a role in this organ development. Okay, we have done a lot of work in this area as well. What are other factors? The molecular factors or the genetic factors, if we look at the 5B, phytochrome B has been shown to play a critical role in the development of potato. We also see constants, we also see CDF1, cyclic DF1, it's a protein, followed by ST-BEL5, the protein that I have worked on extensively in my postdoc, followed also, and the NOX, the master regulator of plant development, and you have SP6A, recently identified another co-regulator cofactor we have identified, another group have identified, that's also a mobile signal for potato. So in addition, you have other factors, you have small RNAs which are known to govern this tuberization phenotype. This pathway is very complex. We thought it was very easy 15 years back. Now 15 years we have been working on now we see that this pathway is far more than what we thought before. It's very complex. There are many factors which are governing this mechanism. It looks very simple, but it is indeed a very complex mechanism. Okay, and many things we have worked. We have been working on this to understand what is this, that how they crosstalk, how they signal, how are the major hormones play in this tuberization phenotype. I will move on and will show you this. So please see, look this. This is the question that we asked long back. What is that signal? Why would this plant, it is now in the mature stage. It is now have lot of mature tubers. And what it happens, please look at this figure. If it senses light, this hook-like stolon, it makes another plant. So for me, this hook-like stolon is nothing but a shoot apical meristem. Just try to understand one fact. This stolon, if it doesn't sense light, it grows a tuber, and that's what the conversion is. This stolon to tuber transition is a dynamic phenomena, is a dynamic process. That's what we believe today. Because we have identified huge number of regulators that can control this process in the early stages of stolon development. And what I'm trying to tell you today, that this is governed by an activator repressor module. Because we have an activator in our hand, we have a repressor in our left hand. And we see how this activator repressor module control this phenomena in the tuber development process. Okay, so let's proceed and see. Tuberization is a reproductive phenomena like flowering, and as I said, florigen and tuber. This is no more florigen, by the way. This is now known. It's a FT, flowering time MR protein, which is now a mobile signal for the flowering. We now know tuberogen, what it is. We have ST-BEL5 and ST-SP6. Two major signals have been discovered in order to understand that what is governing these plants to develop the tuber phenotype. Okay. To cut long story short, I will just try to say one more thing is that this plant makes the plant that we work on. It's a photosensitive. It senses light. It does not make any potatoes as soon as you have a long day duration light. It will only make potatoes when you have a short day light, last duration light. Please remember this point. It will help us to understand. And this is if you put them in long day, we do not get any tuber. So that makes the model organism very fantastic to understand the light and its role in the developmental process. I say it again. This is the review from the pathway. These are all these regulators we have now almost worked out and tried to understand their role. I will just talk about today the 3 mobile RNA that is governing this process, and I will try to come back to this quickly. This is the one that we identified long back, BEL5 protein. Okay, it's known as potato protein, Solanum tuberosum. It's called BEL5 protein, ST-BEL5 I say, and BEL5 interacts with POTH1. These are NOX. NOX are master regulators. Entire plant architecture is governed by NOX, like HOX in Drosophila and NOX in plants. Okay, so these two interact, and we knew this before, long before 2003, we knew that these two interact. So are the other BELs and NOX. And this BEL and NOX interaction is very critical for all types of plant architecture and governing the complete development process in the plant. Okay, so what we did, I will show you few slides from pretty old of my work. We did this BEL5 RNA accumulations in the plant under 2 different light conditions, short day versus long day, and we see huge amount of RNA accumulated in the short day under if you look at the RNA accumulations of BEL5 in this plant, and you see compared to long day, in short day, a lot of RNA is accumulated. So you ask a question, why do you see such a RNA accumulations under a short day photoperiod? Okay, maybe there is something going on in these plants. We overexpress this BEL5 in potato, and what we see that plant makes more number of tubers. That's the message. We overexpress BEL5 in potato. We see plant makes twice the number of tubers. Tubers shapes, size we are not worried about. We see more numbers and more in terms of fresh weight as well. Okay, plants looks pretty normal. If you compare with the wild type, they look very normal. So there was no concern from our side. We knew that if you overexpress, it makes more tubers. That's it. Now, if you reduce, if you make an antisense line, if you knock it down from the plant, what you like to see? If you knock it down, you look at what happens. You have roughly about 30 to 40-fold reduction in the tuber yield. If you overexpress, it makes more tuber. If you reduce it, it makes almost no tuber, you can say. So is that if it's all the lines we checked, we look at the target tuber fresh weight, and we see the tuber yield is reduced in this line. We did a grafting experiment long back, and what we like to show here, this is my overexpression line, and if you graft this line onto a non-inductive potato plant, and if you see, just quickly look at the graph that I did, and this plant, by the way, did not make any tubers. When you grafted this plant made a lot of tubers here. This is after almost 2 months of observations. Sorry, almost 2 months of observations we see. That means something has moved. And that was the puzzle long back. It is no more a puzzle now. This technique is now pretty standard in the plant biology. People do this very routinely now to understand is there any mobile signal. And we did not know what was it. But there were old reports in 1980s and 1985s. We finally really nailed down that what it is. We came down the conclusion. It is the BEST BEL5 RNA which is moving from here and triggering the plant to produce tubers. So BEL5 is a positive signal, for tuberation. It's an activator. It activates the process. Okay, fine. That's the BEL5 all about, old story, right? All these are already published. Nothing new here. You can look at all this. We know where does BEL5 express. To understand this BEL5 expressions, we did a lot of promoter activities, and to identify that where does it express only in the veins, in the phloem, in the companion cells, as well as in the phloem parenchyma, making it more strong signal that if it is a mobile signal, it must be expressed in those tissue types for it to go from leaf to the stolon. That was the time when people showed that. So now I told you that this condition is inductive. If you put the plants in this condition, plant makes a lot of tubers. It's a non-inductive, right? What happens in these two conditions? In non-inductive conditions, you have another protein known as REP1. REP1 level goes high, and REP1 binds to STBEL5 promoter. It is repressing STBEL5 transcription. As a result, whatever STBEL5 is made here is not sufficient to trigger the tuberation. On the other hand, you have the short day. You have ahead of REP1, you have a microRNA 172. MicroRNA 172 cleaves REP1. As a result, BEL5 is free. BEL5 can easily transcribe in this condition, and it can move from here after transcriptions to the target organ and induces the battery of the tuberation circuit. Okay, that is clear now. Okay. I just want to show you just one slide that we have also shown recently, that it is not the 72 alone. There is another microRNA upstream of 72 known as 56. We worked extensively on 56, and we showed that microRNA 56 induces aerial tubers, although the tuber numbers are less, but it produces a lot of all the stems which are likely to grow below ground. All these stolons, they grow from the axils, and plant have a beautiful phenotype. Okay, although the tubers are small in size, but plant have a beautiful phenotype. We are still investigating this mechanism. I'll move on, and I'll try to come back to the main story, that how does this RNA move? Now, this RNA is transcribed here. It can go this way, it can go this way. There was absolutely no clear picture in the field. 2009, the first paper came from UC Davis. They showed that in cucurbits, the pumpkin, there are as many as 16 protein cans binds to an RNA molecule, and it can take it to the up. It has nothing to do with the gravity. Please do not confuse yourself. This is completely a selective guidance mechanism plants have to execute based on its need. Okay, so after this paper, we looked at the potato and we identified those proteins, the orthologs in potato we identified, and then we did lot more work. Okay, so this was the case that these proteins bind to this RNA molecule, and this proteins has polypeptidin RNA binding, RNA recognition motifs, RRMs, and they can bind to it, and this complex known as RNPs, ribonucleoprotein complex, and this complex can go from cell to cell. Imagine it's a long distance transport in case of potato, because it is I said BEL5 expressed heavily in the leaf. After expression in the leaf transcription, there is not much protein is made, very less. We have seen carefully in the leaf that protein is less, and this RNA then moves to the stolon, the below ground organ. What is that? After that, in between. I think I'm missing. Yeah, so I did one more experiment, and I looked at the BEL5 RNA very carefully, and I see that BEL5 RNA has any RNA will have its 5' and 3' UTR. If you look at the BEL5 RNA, it has loaded polypeptidin tracts in both its 5' and 3' UTR. I said, fine, maybe that is making the case for me, in our case, because we do not know which is the protein binding to BEL5, how it is moving. So what we did, we removed those 2 UTRs from the BEL5, and again generated plants and tried to look at its mobility. And we do see there is a very less mobility, absolutely less mobility of the BEL5 RNA, suggesting that those 2 UTRs probably played critical role. If that is the case, now I kept the 2 UTRs intact, put a junk RNA inside, removing the BEL5 from the construct. Okay, that's the beauty of the experiments. You keep both the UTRs intact and put a junk RNA inside. We do see junk RNA is moved, suggesting that my 2 UTRs are indeed playing a major role. We looked at the UTRs, we do see there is loaded with polypeptidin tracts. You have polyUs located in the UTRs. Okay, so these are the old experiments we are just sharing. With the help of those Q-carbit protein that was identified in 2009, we looked at the potato genome. We identified 6 PTBs, polypeptidin tract binding proteins. RNA has polypeptidin tracts, and these proteins bind to this RNA. So we identified 6 of them, and these 2 look like the one that Q-carbit, the UC Davis group identified. And we have again worked on these 2 proteins, PTB1 and PTB6. Now, if you take these proteins and express to the plants, what do you like to see? What is the observations? Okay, by the way, this is the human. This is the human PTB1, and we have looked at that it has RRM1, RRM2, RRM3, and RRM4, the RNA recognition binding motifs where RNA binds to proteins. So what is the function of these PTBs? Why do they bind to RNA? And I said this is the prediction. Yes, we did the RNA EMSA. We showed that it binds to proteins. Where are these proteins expressed? And do they bind mobile RNAs? These are many questions that popped up during the studies. Now, if you look at, I keep saying the polypeptidin tracts. If you look at a particular 3' UTR of PTBs, you can see this is the BEL5, I think. Yeah. So you have a lot of polyUs in between. Okay, and this is where the proteins are likely to bind. We did RNA EMSA, and in RNA EMSA it was shown that the PTB1 binds to BEL5. So suggesting that PTB1 probably they are escorting the BEL5 from leaf to the stolon transport. Now, if we express any of these PTBs, I have a few minutes, 5 minutes? Okay, I'll be done. So if you express PTBs in the plant, if you overexpress PTBs, then now we look at what happens. If you overexpress PTBs, it increases tuber yield. A protein which is a transport protein, which is transporting an RNA molecule, you are expressing in the plant is making more tubers, number 1. Okay, it's also expressing the BEL5. So what is the suggestion is that this protein is basically stabilizing the BEL5 function. Okay, it's making more tubers. It's also like a BEL5, it's an activator. Okay, so we looked at all these. We also looked at the target RNA. We looked at these RNA expressions in the plants and see the conclusions what it is. So STPTB proteins enhance both STBEL5 activity and movement in potato plants. You have another protein, PTB6. If you look at PTB6, I think it's the RNA suppression line. If you suppress it, you see completely no tuber, suggesting that this whatever you got in the overexpression phenotype, in the knockdown phenotype, you have exactly the reverse. So it's suggesting that it is also a positive regulator for potato tuberations. We looked at its expression. It's beautifully expressed in you can look at it, phloem parenchyma. It is expressing in there. You look at how beautiful this protein, STPTB1, how its expression, suggesting this protein is binding to BEL5. It's expressing in the right cell and taking the RNA to the target organ. The potato genome got sequenced 2011. We looked at more bells. There are more bells in the protein. Now we have, I think, 14 bells in the proteogenome. And we wanted to look at 2 more bells which are very close associations with respect to its amino acid sequence with 11 and 29. I can see it here, 11 and 29. These are the two RNA molecules. I already explained to you the ST-BEL5 is a positive regulator. Now I took ST-BEL11 and 29 to look at further because of its close association with BEL5. We thought it will have redundant functions. Now when you overexpress this, the expression pattern of BEL11 and 29, does it affect? We looked at its expression, by the way. So what does this RNA express? And if you look, if you see this is the stolon. I can see from here. This is the short day and this is the long day. Short day is the inductive condition. So this RNA get highly accumulated, suggesting that they may be acting like a BEL5. Okay, so we overexpressed. Okay, you can skip these results. These are also expressing in the phloem parenchyma and companion cells, and we overexpress this BEL11 and 29, the two other transcription factors, their RNA molecule, and we do see they have reduced tuber yield. Unlike the BEL5, they have reduced. That means they are repressing the tuber activity, right? Okay, good. If you suppress it, do they move? Okay, yes, we also looked at, do this RNA molecule move? And we proved by the same grafting technique, they move by a different promoter technologies. We proved that this RNA molecule are also indeed mobile, 11 and 29. Now, interesting thing is, if you suppress this, where is that gone? Yeah, if you suppress this 11 and 29, they make twice the number of tubers. So these two are a repressor, whereas BEL5 is an activator. Now, BEL5, 11, 29, they have pretty close amino acid sequence similarity. You would expect them to have the similar functions, but one is activating the process, other two are repressing the process. Why would you guess it? Why would it happen? Okay, so we looked at its, as I said, these two are also mobile. BEL5 was also mobile, 11 and 29 was also mobile. Now, we looked at its RNA molecule, we looked at its UTRs. They do have the polypeptide tracts. We do not know which protein binds to this. We are now working on this. But at least in the case of BEL5, we know PTB1 binds to it. Now look at this diagram. This is the cartoon on where I'd like you to focus and take your attention here. So this is, as I said, this is nothing but a shoot apical meristem. And this shoot apical meristem, if it senses light, it makes the plant. If it doesn't sense the light, it makes the potatoes. So what is happening? In this shoot apical meristem, we are giving the analogy of that you have three RNA molecules. One is a activator and two are a repressor. 11 and 29 are repressor. And why would you think it will work in this way? Please remember this: before the onset of tuberizations, this organ, what happens before the onset of tuberization? It stops to elongate further. Plant is likely to kickstart the tuberization mechanism. It stops the elongation. As a result, what happens? Between the pith and cortex, the growth initiates, and this growth expands. As a result, you get to see a swelling of this organ, and this swelling expands, and then it accumulates the starch. As such, the swelling starts. You can imagine there is a tremendous change with respect to its radial versus longitudinal cell division pattern. Earlier, it used to be a radial expansion, and it was a longitudinal. Now it will be all radial cell division pattern. So with respect to this 11, 29, and 5, we now explain this fact. These three transcription factors, or their mRNAs, are expressing in these cell types and governing these complete cell division patterns, and thereby making these tolerance to not only it has to target numerous target genes with respect to hormones and many things, right? So you can expect what is going on here, right? And this activator repression mechanism is not new, not that we are first time saying. If you look at flowering with respect to shoot apical meristem, if this is what I said, exactly what is happening here, right? If you look at a shoot apical meristem, and shoot apical meristems, I'm sure many of you may be aware that how shoot apical meristem, the stem cell is governed in the plant cell, how the stem cell number is governed, how does plant accomplish indeterminate growth with respect to maintaining the stem cell between the activation repression mechanism, inhibition mechanism between the ocell and clavata. Okay, so that mechanism is known. So we are trying to understand what is happening with respect to this stolon organ. This is what it is. I can skip. So is the flowering. If you take FT, just one more minute. If you take FT is the flowering signal. If you make one amino acid change in FT, it becomes a repressor. Usually FT is an activator of flowering. One amino acid you change, it becomes a repressor. TFL is a terminal flower homolog. This is an inhibitor of flowering. Okay, so this activation inhibition mechanism in terms of shoot apical meristems, in terms of flower, in terms of potato this stolon development. I said this stolon from stolon to the complete proto development is a complete dynamic process. We are yet to understand that how does it work. Okay, so in summary, what I say, Bell 5, 11, 29 could function collectively as phloem mobile signals in a whole plant network in potato that modulates storage organ development through the process of cell growth activation and suppression in the subapical portions of the stolon tip. Okay, so there is a lot going on there. I can skip this. This is the total summary that we have recently written on review in plant fish, that how does the potato tuber made. You have lot of activators are expressed in the leaf. These activators, I did not explain the measure. Another one recently identified 2011, SP6. SP6 is a co-regulator, okay? It works along with BEL5. And these all signals, it works as a protein. It goes as a protein signal. This one goes as an RNA signal. They all require to go here and turn on the battery of the tuberations mechanism, thereby the tuberations, and work with numerous target genes in this organ. So for me, this is the reaction center. There is a lot going on underground, below ground that we do not know. We are yet to understand. Okay, so my lab is, I collaborate with many people in India as well as in abroad, and these are my generous funding from all of these funding bodies are gratefully and acknowledged, and I thank all of them for their support. These are my papers in last 9 years, or even some of them from postdocs. This is my group in IISER Pune. You are most welcome to visit our institute. And if my concept is whatever I discussed, I like to ask these questions when all experts are sitting here. If the concept of mobile RNA is indeed true, and if it is working that way, these all organs that you see, we are eating all modified, is either modified root or modified stem, or you consider all this now. This was the root before, it became a modified root. Consider this, it's a modified root either. So what's going on here? I'm pretty sure in all these cases, sweet potato we don't work much, but you can imagine with respect to its energy demand, it's so important crop. So we are paying little attention to it, and we're trying to understand, does mobile RNA really work in all these storage crops? Okay, thank you so much. If there's any questions, I will be happy to answer.
Any questions?
Thank you for the nice presentation. Sir, what are the genes responsible for BEL5?
I'm sorry?
Sir, which genes are responsible for the production of BEL5 protein?
What genes are?
Responsible for the BEL5 production.
BEL5 production?
Yes.
Acha. So upstream of the BEL5 we have not worked out. It's BEL5 we identified from a cDNA library. Long back we made a stolon specific library. From library we did a yeast 2 hybrid skin, so we identified BEL5. From the Arabidopsis BEL, we took Arabidopsis BEL as a query, and thereby you pulled out the BEL. These are all transcription factors. They govern many other genes. That's what we do.
Hello, sir. So yes. This BEL5 activator and repressor RNA.
BEL5 is an activator. Repressors are the 11 and 29.
Yeah. Will they three travel together?
Very right questions. I said in summary that it makes sense for me because the subapical stolon is to develop, and I said there is a radial versus longitudinal cell division, complete cell fate change, and these three may be expressing there at a different cell types and governing the different genes with respect to new cell division pattern. Imagine the stolon, which was earlier, it stops elongating, right? It doesn't elongate anymore. It stops just before the onset of tuberization. It stops elongation. Now it swells up. This is the situation, right? It's swelling up, and this is what we think all three RNA are being mobile. Not only all these three. I mean, there are many other pathways are required.
What I'm asking, all three is moving together or what? That's the question. Because if they are moving together, then the same protein must be taking that. Because you didn't find the protein which is taking that.
See, I showed you this plant requires an inductive condition. Under inductive conditions, if you go and test, you will find all three. You will find all three because they are required to go. In the juvenile stage, we don't get to see this, by the way. We see when the plant is becoming the mature and about to start the tuberation. So we see these RNAs are. And these RNAs, they don't, we have looked at their stability as well, and they're highly stable in the leaves. That makes sense that they are the mobile RNAs.
Good evening, sir.
Yes.
Sir, I just have a curiosity that you have worked upon the different activators and repressor, how they regulate tuber formation.
Yeah.
So is it possible that by playing around the genomes using different genome editing tools, etc., we can make tubers above soil? They are normally formed below soil. So is it possible? Because nowadays we are playing around genomes.
I can tell you nothing is impossible. One thing, as you have seen all the great talks before me. Who am I to? But I told you clearly that by simply manipulating one smaller array, 56 microRNA 56, right? We showed the plant develops all the stems not below ground, above the ground, and they are all making tubers. I'm sorry? The test is yet to be tested. Professor Khurana will not let me go like this. He wants an answer. So we are yet to test the nutritional quality. Yes?
Do you know what RNAs are doing? How do they work?
Do I know these RNAs are?
How do they doing?
What are they doing? So in the stolon, what they are doing right now, we do not know. But we have looked at their common targets. We have looked at their all the marker genes, their target, and they all. That means they are there, they are going there, and they are activating those target genes, numerous target genes essentially involved in the hormones. Most importantly, gibberellin. GA is a classical hormone. GA must go down before plants starts the tuberations. And we see the response genes that they target. And they target these RNAs, they make the proteins there. And then they, in tandem, they bind to the target genes. It's called, they make all these autoregulations of their target genes, GA20 oxidase target genes. They bind and regulate the GA level there. So they get translated there, not in the leaf. They all go there. That's the question.
You're not saying that RNA is translated.
RNA is translated at the stolon, below ground, not in the leaf. Not in the leaf. And even if you see these RNAs are translated at a leaf, it's a very, very negligible level. We see probably 10-fold more proteins. Of course, we are yet to see with respect to the antibodies, but we have seen at polysomal level what is happening of those RNAs.
Okay, we have one last question.
So the movement of RNA is through the RNA binding proteins.
Yes.
So when they go to the target, they have the RNA is need to be translated. So how the dissociation of those protein is occurring?
So I encourage you to look at the Plant Cell paper 2009 from UC Davis, the first paper in the field. No, I'll tell you the answer. Answer is this: all these proteins, they do not remain anchored with the RNA anymore. As long as the transport is there, they remain anchored. We have seen the reports clearly describe that at different point of analysis they have seen not all 16 remain anchored. They have got shed off at the transport, but at the target organ 1 or 2 proteins finally still remain. That's what they have shown beautifully. Okay. So they get
Thank you.
Okay, we have one more student question.
It's good, let students ask.
Sir, is there any signal from shoot apical meristem?
There is a signal from is there a signal from shoot apical meristem?
To the RNA, to the proteins to go to take RNA to the stool.
So these PTB proteins, which are polypeptidine tract binding proteins, I did not show their expression pattern. They express. At the same time, when RNA is expressed, if you look at the PTB proteins and their expression patterns, it makes sense for you to understand that they are also in the shoot apical meristems and stems, and they are expressing, right? They are expressing in the phloem tissue, so that they can pick up the RNA molecule and make the complex and then do. We did many molecular assays that they indeed bind and they express.
Because potato tubers are accumulating starch.
Yes.
So usually after flowering, starch gets accumulated in the grains. So I was just thinking in that way.
No, so plants, before they go for flowering, it will induce the tuberation. It will induce the induction of protein.
Like cereal grains, in that case, if the flowering happens,
so flowering, so flowering in cereal clays, as Professor Khurana was describing, in rice, you can see beautiful signal. Now we know which protein goes and signals rice to flower, right?
Okay, I think we'll have to stop there. Please join me in thanking Dr. Banerjee for his presentation.
Yes, so they just get up here with the mic. Okay. The final talk of this symposium is by Professor Michelle Lowen. Yeah, you want this?
Yeah, I'd prefer that.
Oh, yeah.
Okay, we made it. To everybody. Okay, and we'll see if I can do this a little quicker than planned. So is that working there? So I'm going to tell you about a project that I only just really started recently. It's very much a work in progress. And it's regarding a role for the Fusarium graminearum STI2P receptor in mediating the wheat pathogen interaction. So Fusarium graminearum is a necrotrophic fungi, and it is the causative agent of Fusarium head blight in cereals. Whoops, I see the button problem. So that would be, okay, so spores of Fusarium graminearum, they float around in the air. They will land on a healthy wheat head and immediately or shortly thereafter germinate. And the hyphae will grow and infect the rachis, which is kind of like the internal plumbing system on the inside of the wheat head that brings all the nutrients and water up to the head for the developing grains. When the rachis is infected, it turns off that flow of nutrients and water, and the kernels start turning yellow and stop developing and eventually shrivel up and die. And this leads to dramatic decreases in yields for the farmers. These are just some Fusarium spores. So the other problem with Fusarium is that, or graminearum, is that it also deposits mycotoxins. In particular, it deposits deoxynivalenol, which is known as DON or vomitoxin. And while it's part of the infection process that allows this to be a necrotrophic fungi, it's also bad for animals and humans who consume the DON-contaminated grain. It leads to horrendous vomiting, and people have died of dehydration and whatnot from it. So it is a problem. Historically, we've not had Fusarium head blight in Canada, but because our main wheat growing area is in the western provinces, Manitoba, Saskatchewan, Alberta, where it's quite cool and relatively dry. But with climate change, we're seeing more humidity and warmer temperatures generally, and with that we're seeing a significant increase in the occurrence of Fusarium head blight in Canada, most notably over the last 10 years. So to address this and a variety of other issues with wheat in Canada, the Canadian government in 2012 initiated what we call the Canadian Wheat Alliance. And it brought 53 principal investigators together from 4 or 5 different institutions with the mandate to improve the yield, sustainability, and profitability of wheat. And they asked if I would participate in that, and I said, sure, maybe I can help with Fusarium. But there's other members looking at all other aspects of improved wheat in Canada. Okay, so on to the molecular. So this project that I just initiated recently in this context is related to the STI2P receptor of the graminearum pathogen, which is a G protein coupled receptor. It is STI2P and its close homologue STI3P are the pheromone mating response factors in fungi generally. And the way this works is that you have two mating cell types, MAT-A and MAT-alpha. MAT-A cells secrete the A factor pheromone, which is a short 10 to 12 amino acid peptide. This will bind selectively to the STI3P receptor that is only expressed on the MAT-alpha cells. MAT-alpha cells will secrete a different pheromone, the alpha factor, which is just a different peptide, which will only bind to the STI2P receptor, which is only expressed on the MAT-A cells. So in both instances, binding of the pheromone to the receptor leads to classic signal transduction, including stimulation of a heterotrimeric G protein, so you have dissociation of the alpha subunit from the beta gamma, and stimulation of a mitogen activated protein kinase cascade, which ultimately leads to modulation of gene expression and cell cycle arrest. Once the cells have arrested, they're then poised to undergo the second portion of this process, which is the actual mating events, where you have projection or smooth formation. So this is where the cells deform and start growing toward each other. Cell fusion, so that's the cytoplasm and nucleus actually fuse, and ultimately a diploid cell formation. So this is an example of classic GPCR signal transduction. You have one ligand stimulating one receptor, leading to stimulation of one pathway. So I've been studying ST2P ever since I left Gobin's lab 17 years ago. And most recently, I've been interested in the idea of whether there's the possibility of biased signaling in ST2P. And what biased signaling is a newer concept in the field of GPCR, where for a single receptor, you can actually have different ligands. And those different ligands will actually stimulate different signal transduction pathways through that same receptor, leading to different biological outcomes. So in terms of the work, I did this all pre-wheat. We obtained evidence of a second lower affinity pheromone binding site on the ST2P receptor that actually seems to stimulate initiation of the second part of the mating pathway. At the same time, we also obtained evidence that arrestin may be involved in mediating this effect, although we haven't yet identified what pathway this might be signaling through. So that's what I was doing before, and just keep that in the back of your mind. I'll come back now to Fusarium graminearum. So about 18 months ago, in 2015, late 2015, this paper came out in Nature by Antonio Di Pietro. He's a Spanish fellow, and he's working on Fusarium oxysporum, which is a little different than graminearum. It's a soil-borne fungus, so the spores remain in the soil, and when they germinate, they grow toward the roots. And what Di Pietro showed is that the roots of this tomato plant actually secrete a peroxidase enzyme, the catalytic activity of which stimulates ST2P, leading to transduction down a different pathway, a cell wall integrity pathway, ultimately eliciting the chemotactic response toward the roots. So I was so excited when this paper came out. Here it is, evidence also of biased signaling for ST2P. But more importantly is not the results itself, but the questions that any good finding yields. You find this, you make a discovery, there's 10 more questions. And so these are the black boxes. So first and foremost in my mind is, what is the nature of the ligand that the peroxidase is producing that's going to be stimulating that receptor? And what is the effector that's mediating the signal from the receptor down to the CWI pathway? Does this extend to other fungal species that infect other plants? For example, graminearum. Does the wheat head secrete a peroxidase? Is there a CWI pathway? Chemotaxis toward what? If the spore is already sitting there on the wheat head, what would be the point? What about getting away from plants? A human fungal pathogen, Candida albicans. Humans also do produce peroxidase as an immune response, so it's a possibility. And in terms of Cerevisiae, is the CWI pathway potentially mediating the biased signaling that we're seeing in the mating response? So lots of questions there, probably to see me through to retirement, I hope. So we wanted to investigate Fusarium graminearum, so we set up a chemotactic plate assay. Essentially, we have just a layer of agar, with water agar with spores embedded in it. We have a scoring line and two wells, one on each side of the line, with a test compound that we're going to test chemotaxis toward, and then a solvent control. We let it sit for about 12 hours. We count up the number of hyphae that grow toward the test compound compared to the solvent control. And from that, we can get a chemotropic index, where a positive value means chemotropism. So the first things that we tested were nutrients. We assumed that we were doing this more as sort of a positive control. Oxysporum showed a strong nutrient response. Graminearum did not, much to our surprise. There was no significant, to these basic carbon and nitrogen sources, there was no significant response. Except methionine, which is interesting, and I'd be happy to talk to anybody about that at dinner. So we were concerned that maybe the system wasn't going to work, and so then we tested the pheromone as another positive control, and this time we did see a strong response to the Graminearum pheromone, not the Cerevisiae pheromone, showing that it is a specific response, so we felt we could move forward with more experiments. So of course peroxidase was the main target, so we just bought some from Sigma, horseradish peroxidase, so we could get on with it quickly. And indeed we saw a strong response to the peroxidase. This was eliminated when we incubated it with proteinase K, or if we boiled it, showing that it really is the enzyme. And when we added a peroxidase inhibitor, this also eliminated the activity, showing that it is the actual peroxidase activity. You need active catalysis there to get the effect. So what about wheat? Going back to the wheat issue. So here, as you can see in this picture, we grew some wheat, and we wanted to see whether there was chemotaxis toward the wheat head. So we tipped the wheat plants over and put the wheat heads into our plate with a bit of water to make contact, and then just did the same thing. We tested 3 different wheat varieties. Roblin is quite susceptible to FHB, Sumai and Wuhan are resistant, but that didn't matter. We saw chemotaxis regardless of the resistant state. So this was promising. It seems we are seeing chemotaxis to the wheat head. We then obtained what I refer to as a secretome, or an exudate, where we took the wheat head and we put it in a test tube of water and left it for 48 hours. And anything that secreted out, we called that the secretome. The postdoc who was working on this said it feels like alchemy, Michelle. This is just alchemy. But anyway, it was working. So we concentrated that secretome, put it in our assay, and again we see even a concentration-dependent response to this exudate that is sensitive again to the peroxidase inhibitor, suggesting that there is peroxidase in this exudate. We subsequently tested for peroxidase activity in the exudate. This was just an in vitro photospectrometric assay, so we did see peroxidase activity. When we ran it on a gel, we saw quite a few protein bands, and when we looked at these by mass spec, one of them came back encoding for a peroxidase. So, and that's as far as we've gotten with that so far. Going back to the fungus, we still needed to link it to ST2P. To do this, we knocked out the ST2P receptor. So we made a line without ST2P. And you can see that in all the instances where we saw strong chemotactic responses in wild type, deletion of ST2P did indeed eliminate the effect, linking the effects to the receptor. And that's it. That's as far as we've gotten so far. So I feel that, I mean, that's about 6 months worth of work that I've shown you there. But I think we've made a little bit of progress on the black boxes. We know that the wheat head is secreting a peroxidase. It's stimulating ST2P, and this is leading to chemotaxis. But still lots of unanswered questions. I'm mostly interested in investigating what the actual ligand is. So that's what's coming up next. I had hoped I'd have some data on that for you, but perhaps my expectations were a bit high of my graduate student, so sorry about that. On the applied side, so what's the relevance of all this? On the one hand is the possibility of new antifungals. So agriculture needs new antifungals. All the existing ones essentially work by the same mechanism, which is modification of the plasticity of the cell wall. And resistance is developing, and as resistance develops, farmers are applying more and more to their crops, and we're getting accumulation and toxicity in the fields on other organisms, and there's talk of carcinogenicity on humans, so we need new antifungals. And the possibility exists that ST2P might be a really good target in that it presents a novel mechanism. So it's working by a different mechanism than all the existing antifungals. We want something that's more efficient so that we can apply less to the fields. And it's also specific. Only fungi have ST2P, so that even if the antifungal accumulates in the field, it may not affect more than just the, it should not affect more than just the fungi. And last but not least, the breeders really like to breed better lines, and knowing that peroxidase contributes to infection, if we can potentially breed lines that secrete less peroxidase, can we across the board decrease fungal infection of wheat? So we'll be working on all of those aspects going forward. Quickly, I just want to thank in particular the two Poojas. So they were two PhD students that I recruited from here in India. Pooja Chudhari did all the Saccharomyces cerevisiae work on the biased signaling, and Pooja Sridhar made the ST2P knockout in Graminearum. And I am potentially looking for a new graduate student start next summer, so if your name is Pooja, let me know. Okay, and this is the last slide I'm going to put up today. So I want to thank Goban, who inspired a career-long love of G-protein coupled receptors, which is why I started this project earlier this year. But perhaps he also contributed more to my lifelong love of science. I, in fact, have known Goban my whole life. This was a picture my father took. That was my father in the slide yesterday. He was a postdoc for Goban 70 to 72. And I was a postdoc for Goban 98 to 2001. Here is a slide, or a picture my dad took. He labeled the slide HGK and Michelle, August 1971. So I'm 1 year old. And here's Goban. Sorry you can't see his face and with the lighting, but you can still see a hint of his ears. So you see the ears here. Those are definitely Goban's ears. So that's it. And I'll just leave you with this last thought. Goban said to me on a number of occasions when I worked for him, good science is all about the people. And I really didn't get it when I was a postdoc, because it seemed to me it was all about 24/7 at the bench getting results and papers. But now 20 years later, I do get it. And it is all about the people, and I think that's why we're all here today.
Thank you so much, Professor Loven. Can we have one or two questions?
How about a dinner?
Okay. So yeah. So the final poster awards. May I now request Dr. T.R. Sharma, Dr. Uttam Raj Bhandari, Dr. Ranu, and Dr. Arora, Dr. Dieter Sohl to please come on the dais and give away the best poster awards. And may I now request Dr. Loven to please do the honors of announcing the poster awards.
Sure. I'll use this one. So, sorry, just what a...
Yeah, so you just need to announce it in this sequence.
And also tell a little bit about the quality of posters.
You will? Or me? You. Oh, yeah, I was going to.
And then the poster numbers.
And where it stops here?
No, it starts here and then there
are there's
16.
Okay. Okay, so Dr. Oh, thank you. So, got it done quick.
Everything right.
Okay, so we're going to do some poster awards. We have some distinguished members here to give out the awards and shake some hands. So a number of us, 5 or 6 or 7 of the HGK associates evaluated posters last night, and I think we were talking about it later, and we were all quite impressed with the quality of the posters presented here at this conference. So yeah, congratulations to everybody. It was a real eye-opener about the research going on here in India. Okay, and so with that, I'm going to start reading down the list. So, Best Poster Awards. Poster number 2, Shruti Gupta.
From JNU.
From JNU, in case there's 2.
Okay, so she's
not here.
Yes.
Okay, Kirti Gupta from CSIR. Still not here. Okay, this will be quick then. Poster number 17, Ankush Garg from INST, Mohali. Aha, a local student. Great.
So it's actually the cash award which we are giving to the best posters and some books also. Where are the books? So it's going to come later. Some cash and some books. Yeah.
Okay. They're just getting their act together. We got it down now. Okay, next poster is number 20, Harsimran Kaur, INST, Mohali.
Yes, sir.
Okay, next poster, interestingly, is number 22, Pooja Sharma from INST. Yeah. Sure. Come here. Congratulations. Number 38, Navneet Kaur, or Kavar.
From Nabi Mohali.
From Nabi. Next is number 90, oh my goodness.
Ujjwal Kumar Trivedi.
From Marwadi University of Rajkot. Aha. I see.
Oh, I'm sorry.
Technical difficulty. Nice. Okay, next is number 110, Satya Narayan Patel from CIAB. Is that a 9 or a 4?
64.
Following is number 64, Swapnil Shinde, CDFD. Should I say anything about that or not?
Okay, now this one.
So I'll just carry on, okay. Okay, next, looks like we're starting back at the bottom, number 23, Nistha Sharma, Nabi. Next is number 44, Tassina Akhtar. Punjabi University.
Come here.
Next is number 77, Rajni Ved.
Shulni University, Solan.
Yes,
next is number 49, Sheesh Marath from ICAR, ICAR. Perhaps not here. Okay. Number 41, Afsana Parveyan, Nabi. Okay, number 52, Anjali Purohit, CIAB. Number 28, Shivi Tyogi, Punjab University.
She's here. Yeah.
Oh, quick.
Yes. Okay.
Okay, and last but certainly not least, R. Jaswal Nabi. Or can you?
Yeah, R. Jaswal. Thank you so much, dignitaries. Now we'll start with the conclusion session. The floor is yours, Dr. Rano.
First, I have an announcement to make. You would like?
No, yeah, that's one thing. You said thank you to everybody first.
Yeah, okay. We have a couple of announcements to make. One is that Dr. Raj Bhandari is going to give the Qurana lectures tomorrow. And so you're all welcome to come. Because of time issue, I couldn't go into much about Govind's personal side, but tomorrow Tom has been with Govind forever, so you will get a little better taste of it, about his personality, about his professional career, and how he viewed different things, and his hobbies and other things. So that's the announcement. And next is both Tom and I team in USA would like to thank the 4 directors, the Dr. Sharma, the Dr. Ganguly, Dr. Arora, and I don't know the name of the ICER director, but whoever it is. I haven't met him. So, director of ICER, for being partners with us in bringing this truly special symposium to the soil of the birth of Govind, Punjab. And we are really sitting in Punjab. And we are not in Chandigarh. So the second thing is that Team India consists of not just the directors, Ajay, Susmiya, Aziz Paul, Dipinder Kaur, and many other people who have worked behind the scenes, including the IT people who managed to bring Shri Ram live from NIH, and all the other folks who have participated in making this symposium such a spectacularly successful, and all the visitors who have come from overseas have told me that the host has done a fantastic job, and they have taken care of them to bring them here, take them back to the hotels, and very efficiently. And they are all truly grateful to you guys. And also, I would definitely take this time to say again thank you to Khurana Associates who managed to make it. There are a bunch of people, believe me, some last minute things happen. I mean, Ricky had an accident with a bus day before, and she could come. And some other people got sick, and Santano got cold. So there are few, almost I think 6 people who couldn't make it. They had planned to come, so we appreciate their cooperation, their friendship, their help in bringing this thing alive. So with that, I think I'm going to give this microphone to Tom, and we have a very beautiful surprise for you guys.
So this meeting has really been about as successful This meeting has been about as successful, really, and as enjoyable as we could have hoped. Thank you. Really, beautiful job done. Everybody comfortable, and I think everybody happy. Thank you. Thank you. Thank you. Now, what Rajendra was offering, saying was the following, that the first meeting, series of this meeting was held in Cambridge, England in 1985. At that meeting, Govind and Esther were both there. And the organizers of the meeting, there was Mike Gade, who was here and left this afternoon, and Dick Walker. What they did was to have Govind and Esther sign a document, personally sign a document. And it turns out that Mike had found before he came here that there are 3 of those documents left. And we thought what to do with it, and they said that we should distribute it to some people we thought deserves it here in this setting, with the hope that these documents will be maybe displayed at a particularly visible place, like Nabi here, for instance, would be downstairs, entry hall somewhere, so that the people who come in can see it, students, postdocs, and everybody who works can see it, and get inspired by what they say. And so what we would like to do is to really, we thought the three people who, or the three institutions we should leave it with would be, of course, Nabi here, and Dr. Ganguly's place, and Dr. Arora's place. So if
we wish that we had another one for the ISER, but we are really sorry about that. But these are paintings by artists in England, signed by both Govind Khurana and Esther. And also, we want to emphasize this point. This painting will not go into the director's office or in any hidden hallway. They will be on display for students, postdocs, young people, so they can have inspiration. Again, this is a very important point. So I'm going to unveil it, and so you can see it, and then we will call each one of the directors. So they'll be nicely framed. All right, so Dr. Sharma, can we request the gentleman? Okay.
Both we.
Okay, that's fine.
There's Katilda, Rakshit, and Rikesh, who was behind the scenes.
Can I come like this? Thank you very much.
No, but I think you should bring your
village
team.
Please.
Okay. She's so happy.
Thank you.
So, actually, he brought 4, but one was for me because I wasn't there.
That's it.
That's it from our side. If Dr. Sharma, the floor is yours.
I don't have much to say except to thank all the Khurana associates for choosing Nabi as a venue for this very important symposium. In fact, it's a treat for all of us, for the students, for technical officers, and also for the scientists. And let me tell you, this is the first ever symposium we organized in this very hall. It's a very new institute, a brand new hall in which you are sitting. So we are very lucky, and on behalf of all 4 institutions, I would like to thank you all. And I wish that such symposium would always be organized in this particular hall, so that we should always be benefited from this scientific discussion. Thank you very much, and goodbye.
Final announcement. For dinner, we are going to go to Hotel Shivalik, and the transport committee has made the arrangements. So the buses will be available downstairs at 6:45 p.m. So they will take you to the venue. After having dinner, they will again drop you back to the to Nabi campus. Yeah, thank you. Very good evening.